Following are the learning outcomes that are expected na makuha ng mga students after this discussion. First is to discuss enzymes and the major functions within the biological system. Next is to explain the different terminologies related to enzymes. Classify enzymes based on the reactions that they facilitate, the catalytic activity of enzymes, analyze the relationship between enzyme and also substrate, differentiate principles of catalysis and specificity, and then catalytic mechanisms as we mentioned earlier. Construct a graph illustration of the relationship between enzyme catalyzed reactions. Identify the relationship of substrate concentration and the reaction rate through mathematical equation. And finally, solve problems related to Michaelis-Menten kinetics and graphical illustration on the different types of enzymic reaction. As an introduction to enzymes, basically enzymes are proteins, and these are the ones responsible in increasing the rate of chemical reactions that happen in the body. So when we say enhancing the rate of chemical reaction, that implies that enzymes can make the reaction process go faster. That is, in a catalytic process, a certain substrate, also known as the reactant, through chemical reaction is being converted into a product. So for this reaction to happen in a faster way, enzymes are needed. That's why enzymes can increase the rate of reaction. Now, you have to realize that enzymes form a complex with substrate through temporary covalent bonding, and through temporary covalent bonding that causes dissociation after the reaction took place, making enzymes unchanged after the overall rate of chemical reaction. And take note that the catalytic activity of enzymes depends on their native conformation. This tells us that the activity of enzyme depends on the proper folding of the proteins, that is the primary, secondary, tertiary, and quaternary structures. And as we said, enzymes target the substrate for the substrate to be converted into products in a faster means. Most enzymes are proteins, but you have to realize that there are some exceptions, like for instance catalytic RNAs. So yung mga catalytic RNAs, these are enzymes basically, catalytic RNAs that are enzymes pero hindi naman sila mga proteins. And usually enzymes don't require other entities for them to be functional. But you have to drink this in that there are enzymes that for them to be functional, some non-enzymic components are needed. So for example, for the non-protein or non-peptide component, we have cofactor and coenzyme. The difference between the two is that ang cofactor, it contains one or more inorganic ions, while coenzyme contains metals, right? Metals or metallo-organic molecule. So as examples, we have cofactors shown on this table. You have to memorize this because this could emerge in the quiz. For example, zinc ions are needed for the activity of carbonic anhydrase and also carboxypeptidases. Magnesium ions for EcoRV and also hexokinase. Nickel for urease, and so on. So ang point dito is, for this to be functional, cofactors or metals ions are needed. What about coenzymes? Coenzymes could be categorized into two. This could be co-substrate and prosthetic group. Co-substrate acts as transient carriers of specific functional groups, and these are usually derived from vitamins and organic nutrients. Whereas prosthetic group is the one that is tightly bound to the enzyme protein. So tightly bound, which means na kadikit na talaga niya yung enzyme. So for the example of coenzymes, usually coenzymes are the ones derived from vitamins. And often these coenzymes come from the vitamins like thiamine, which is B1, riboflavin B2, niacin B3, pantothenic acid B5 with their corresponding coenzymes, in which for TPP, it is responsible for aldehyde transfer, for FAD from B2 for electron transfer, and then B3 for nicotinamide, we have hydride ion, and then B5 naman for acyl transfer. Pyridoxine is B6, folic acid is B9, cyanocobalamin is B12, and then biotin is vitamin H. Okay, vitamin H. So with this, meron tayong mga coenzymes din derived from these vitamins, such as pyridoxal phosphate, tetrahydrofolate, and then coenzyme B12, and then biocytin. These are important for the transfer of these groups. Again, you have to memorize that. Moving on, we have the term holoenzyme naman. Holoenzyme, this is a catalytically active enzyme together with its bound coenzyme or metal ions, in which the apoprotein is the protein part. Apoprotein is the protein part, also known as the apoenzyme, and when combined to a cofactor, it becomes holoenzyme. Whereas kapag inactive pa siya, zymogen naman ang term. So those are the terminologies related to enzyme. Moving forward, let's have the nomenclature. Usually, marami tayong mga naririnig na terms related to enzymes. And for us to be guided, let's present those possible variations. Ang pinaka-common na nomenclature for enzymes is by using the suffix ase. Again, using the suffix ase. So with this, malalaman natin kung ano ba yung function ng isang enzyme. So, for example, we have dehydrogenases. Dehydrogenases are the ones that provide removal of hydrogen. So, obviously, there could be oxidation reaction here. Proteases naman are the ones that target proteins through hydrolysis, a breaking down of peptide bonds. Remember, breaking down of peptide bonds. And then, isomerases for rearrangement. Next, another way to name enzymes that is based on modifiers. So for example, we can have the name enzyme based on the substrate that it targets. For instance, xanthine oxidase, wherein this enzyme facilitates oxidation of the purine xanthine. Pwede ring source kung sino ang nagsesecrete, like for instance, pancreatic ribonuclease, in which ang ribonuclease, it comes from the pancreas. Regulation, like hormone sensitive lipase, and then feature of its mechanism of action, like cysteine protease. We can also have nomenclature like alphanumeric designations, like for instance RNA polymerase 3, DNA polymerase 1, DNA polymerase 3, RNA polymerase 1, and so on. O kaya Greek alphabet, like protein kinase C beta. And then some enzymes retain their original trivial names, like for instance for pepsine, trypsin, and also chymotrypsin, all of which are responsible for hydrolysis of proteins. Given that there are numerous nomenclatures about enzymes, the problem that could emerge there is the ambiguity. Pwede magkaroon na misunderstanding on what enzymes are being referred to. So to be able to make nomenclature systematic, uniform, and also universal, the International Union of Biochemistry and Molecular Biology developed a system in classifying enzymes. And that is by giving codes that is coded from 1 to 6. And these are the 6 major classes of enzymes, starting from oxidoreductase, transferase, hydrolase, lyase, isomerase, and finally ligase. Mind you that the sequencing is rigid. Hindi dapat magkapalit-palit ang sequencing ng class number kasi naka-base sa number sa sequence kung anong klaseng enzyme siya. So for example, kung 3 hydrolase yun, hindi pwede maging oxidoreductase. Okay? And on the second column of the table, this is the column that tells us the type of reaction that they facilitate. For example, for oxidoreductase, we have redox reactions, transferase, transfer of a group, hydrolase, hydrolysis, lyase, addition and removal to form double bonds, and then isomerization, molecular rearrangement, and then we have ligation through ligases. So usually ang napagpapalit ng students dito ay ang 4th at ang 6th kasi para silang L. Para matandaan nyo, lyase means separation, and kapag naman ligase, merging, merging. So usually sa relasyon, nagkakahiwalay minsan, nauuna yon, at sa uli magkakabalikan din. Sa haba man ng prosesyon, kaya haba-haba man ng prosesyon, sa simbahan din ang tuloy, magkakatuloy na sila. O, di ba? O, ganun ang palatandaan. So, oth, lil. Okay? Oth, lil. Okay? Next, for an example, para magets natin, usually coded nga ang name ng enzyme. Like for instance, in this given sample, 2.7.1.1, in which the first numeral pertains to the transferase enzyme. Okay? So let's now discuss individually these classifications. So first stop, we have oxidoreductases. Remember that these are the ones responsible for redox reactions. Examples are dehydrogenase, oxidase, reductase, and also catalase. Next, for transferase, these are the ones that facilitate transfer of functional groups like transfer of acetyl, methyl, and phosphate. In particular, please note that there is a specific name for transferase enzyme that allows transfer of phosphates. At yun ay ang kinase enzyme. Again, kinase. So yan ang responsible. And then to allow understanding on transferase, let's have these common examples like acetyltransferase, promoting transfer of acetyl group, methylase, methyl group, kinase, transfer of phosphate, and polymerase. Particularly ang polymerase, responsible ito for DNA, and also RNA synthesis. DNA and RNA synthesis that provides transfer of nitrogenous bases. That provides transfer of nitrogenous bases to allow elongation of the strand for both DNA and RNA. You will further understand this once you proceed on nucleic acids part. So, mag-focus muna tayo sa transfer of acetyl, methyl, and phosphate. So, for example, in transfer of acetyl group, we have here the given substrate in which after acetylation reaction, there is addition of acetyl group. And remember, acetyl group is a substituent, carboxyl substituent having 2 carbons. So, 1 and then 2. Next, for methylation naman, you expect here that there is addition of methyl group, like for instance, from the nitrogenous base cytosine, that through methylase enzyme, nagiging thymine siya. So notice that through methylation, there is addition of methyl group here, addition of methyl group. And then finally, for phosphorylation is made possible by kinase because there is transfer of phosphate. So you can check that on the product after catalysis by the protein kinase, may addition of phosphate. Next, proceed naman tayo sa hydrolases. Regarding hydrolases, these are the ones that catalyze hydrolytic reaction, breaking down of bonds due to the presence of water. Usually, ang mga substrate dito, again, ang mga substrate ay yung mga may amide functional group and also ester functional group. Again, yung mga substrate ay merong amide at ester functional group. Next, for instance, we have proteases. For proteases, we have HIV protease and caspase. Nucleases naman, these are the ones that break phosphodiester bond. Ay break phosphodiester bond or phosphodiester linkage that would break nitrogenous bases, or that would remove nitrogenous bases. Yeah. Okay. So next, regarding nucleases, as I've said, nucleases are the ones that can cleave off phosphodiester linkage. And phosphodiester bond could be seen in nucleotides. In nucleotides, once they are connected all together. You will further understand this once we discuss nucleic acids. So stay tuned, madidiscuss natin to lahat sa nucleic acids. And then we have phosphatases that can catalyze dephosphorylation. So sa dephosphorylation, there is removal of phosphate group. This is the opposite of kinase. Kasi sa kinase, you can expect that there is an addition of phosphate group doon sa substrate, dito removal. Ayun yung difference. Again, removal dito, sa kanina sa kinase addition. So example, you have calcineurin enzyme that is being blocked by the immunosuppressant cyclosporine. We also have liases. Liases are the ones that catalyze the cleavage of carbon to carbon, carbon to oxygen, carbon to sulfur, and carbon to nitrogen bonds that causes formation of additional bond. And then examples are decarboxylase and aldolase. Isomerases provide molecular rearrangement such as rotamase, protein disulfide isomerase, and then we have epimerase and racemase. And for the last, we have ligases. Examples, or these are the ones that allow joining of two molecules. Examples, we have peptide synthase, amino acyl tRNA synthetase, DNA ligase, and also RNA ligase. So those are the different enzymic classifications. So moving forward, let's proceed on the different mechanisms of enzyme action. Please realize that enzymes are the ones, I repeat, these are the ones that allow conversion of a substrate into a product sa mabilis na paraan. So nangyayari yon kasi ang enzyme ay merong special pocket called the active site that allows binding of the substrate or the reactant. So such binding of the substrate or the reactant on the active site can cause different reactions or forces to occur, leading in the formation of a product in time. So that is enzyme, then substrate, enzyme-substrate complex, and then finally, the product will be formed. So yun yung ibig sabihin natin dyan. So to be able to understand this further, let me repeat that there is the active site present in the enzyme where substrate will bind, leading to formation of enzyme substrate complex, and through chemical reactions here there is enzyme product complex, and finally there is dissociation. So in dissociation, product ay hihiwalay sa enzyme. That is why product is already formed in a higher rate or faster way, and then enzyme can be used again for another round of reaction. The function of enzymes is to increase the rate of reaction, not equilibria. And when we say reaction equilibria, it means that there is no net change in the concentration of reactants and product. So to be able to express graphically the activity of enzymes, let's discuss this graph or this diagram. This diagram depicts the reaction coordinate diagram of an uncatalyzed reaction. Una sa lahat, pag sinabi nating uncatalyzed reaction, ibig sabihin yan, conversion ng substrate o ng reactant papunta sa pagiging product pero walang enzyme. Kaya nga, uncatalyzed, walang enzyme. Nabanggit natin kanina na oo, nagagawa ang product through chemical reaction, ngunit pag walang enzyme, mabagal siya. Ay mabagal. Pero may product. Pero mabagal. Okay? So ngayon ipaliwanag natin bakit mabagal ang formation ng product kapag walang enzyme. So this is how it goes. First stop, kindly realize that on the vertical or y-axis we have the free energy or the Gibbs free energy. Gibbs free energy. And the reaction, this is the reaction progress, meaning yung pagbabago dun sa reaction. And note that we have this, the gives free energy for the substrate at ground state, and then the product at ground state. So dito yung substrate, ito yung substrate, and then we have here the product. Now, ano yung important na dapat malaman dito? First stop, yung value, yung estimated value ng free energy ng substrate at ng product. You can see here that higher gives free energy is observed in the substrate and lower in the product, making us conclude that lesser energy is observed in the product, making it a more stable form, ay more stable. Sa substrate, mas maraming free energy, so mas malikot siya. Now, if we intend to convert a substrate into a product, energy must be increased. Kailangan niyang tumaas as the reaction progresses. Kaya you can see here, gradually, na tumataas ang level ng Gibbs free energy until the climax is being reached. So, ang tawag sa climax na yon ay transition state. Ang transition state, this is defined as the molecular moment. This is defined as the fleeting molecular moment. Again, this is defined as the fleeting molecular moment in which the reaction could either proceed or not proceed. So ito yung climax na dapat nating abangan kung ang reaction ba ay tutuloy pa kanan o babalik siya pa kaliwa. So para can you follow? Again, kung matutuloy pa kanan, pa right, o kaya hindi. Now, para maging product ang substrate, kailangan ang transition state ay ma-overcome. So this must be overcome. Again, this must be overcome. Kailangan malagpasan itong climax. Ngayon, pag hindi ito nalagpasan, hindi magiging product ng substrate. Okay? So, for example, na-surpass o na-overcome ang transition state. Such overcoming would cause lowering of Gibbs free energy until such time that the product will be formed. And notice that product has again lesser free energy, making it more stable. However, you can see here that there is a significant difference in the level of Gibbs free energy of the substrate and also Gibbs free energy of the product. Ang point dito is bumagsak at meron siyang corresponding value. It's a negative value, okay? Kasi decrease. Ang tawag doon ay exergonic reaction. Again, ang tawag doon ay exergonic reaction kasi nag-release ng energy. Okay, nag-release. And then, ano pa yung important dito? Ang important din dito, class, is yung na-consume na difference para ma-overcome ang transition state. So, anong tawag natin sa range na ito? Ito yung energy kasi na ginamit upang ma-overcome yung molecular moment. So, ang tawag dito, everybody, again, ang tawag dito ay activation energy. Again, ang tawag ay activation energy. Again, this is called as the activation energy. So, ito yung dapat malagpasan, ay activation energy. So, overall, yan ang reaction coordinate diagram kapag uncatalyzed reaction. Ulitin natin para ma-summarize. Again, ulitin natin. Sa uncatalyzed reaction, burahin ko. Sa uncatalyzed reaction, kailangan malagpasan ang range na ito. Anong tawag dito? Very good. Activation energy. Again, activation energy. And then, anong pinagkaiba ng amount ng energy sa substrate at sa product? Sino mas malaki? Very good. Substrate. Mas mababa ang? Okay, product. So, anong mangyayari? Positive or negative? Yes, negative effect, negative difference. So, kapag negative, anong tawag doon? Yes, very good. So, at least nakakasunod, exergonic. And, anong tawag sa transition state? Yung ating analogy, ano yon? Okay, climax. Ay, climax. Kailangan yan ang ma-overcome. Okay? Yan yung meron o walang enzyme. Okay. walang enzyme. Walang enzyme. So ngayon, paano natin ma-appreciate na ang enzyme ay nagpapabilis ng conversion ng substrate at sa product? So i-discuss natin itong figure na ito. So may dalawa na tayong graphs na makikita sa reaction coordinate diagram. Yung walang enzyme at merong enzyme. Yung itim, yung black, ito yung walang enzyme. Yung blue, may enzyme na. Tingnan nyo mabuti, una ang substrate at sa product. Ano nangyari? Parehas lang sila. May enzyme man o wala, parehas ng level, lalo na ng product. Okay? O ano pang mapapansin? Negative din, yes, endergonic din. Exergonic din. So negative siya. Mas stable ang product as the reaction progresses. Anong pagkakaibang napansin nyo? Mas mataas. Anong napansin nyo sa transition state? Transition state, yung climax ng walang enzyme, uncatalyzed sa catalyzed. Mas mataas ang transition state ng uncatalyzed. Meaning, mas malaking enerhiya, mas malaking activation energy ang nakokonsume kapag walang enzyme. Pero pag may enzyme, mas mababa. So, taken all together, anong realization natin dito? Ang realization is that enzymes lower activation energy. Again, they lower activation energy. And by lowering activation energy, what can you conclude? They make the conversion of a substrate into a product go faster. Napapabilis nila. Okay? So yan ang tatandaan nyo. Napabibilis kasi napapababa ang activation energy. Do you understand? Nice. Okay. So, dagdagan natin just to trigger your minds on the succeeding topic that we will discuss. Ngayon, anong tawag sa energy difference sa pagitan ng uncatalyzed at ng catalyzed reaction? Di ba nung nag-drawing tayo kanina, naalala nyo, di ba? Meron tayong graph sa catalyzed at meron tayong graph sa, may graph tayo sa uncatalyzed at meron tayong graph sa catalyzed. So napansin natin na merong energy difference, yung nawala, yung natipid, yung natipid na enerhiya. Ang tawag sa natipid na enerhiya, binding energy. Again, ang tawag dito ay binding energy. O sulat nyo sa notes nyo, pag sinabi nating binding energy, this is the energy difference between the transition state of uncatalyzed and also catalyzed reaction. Catalyzed reaction. And take note, the higher the binding energy, the higher the binding energy, the faster the reaction is kasi bumaba ang activation energy. Ulitin natin. So kapag mataas ang binding energy, tanong, mas malaki o mas maliit ang natipid ng may enzyme? Okay, mas malaki yung natipid. So pag mas malaki yung natipid na enerhiya nung gumamit ng enzyme, kumonte yung activation energy, bumaba siya, bumilis ang reaction. Scientifically, inversely proportional. Okay? So that's for enzyme catalyzed reaction. So yan ang nangyayari. Let's now proceed on the catalytic power of enzymes. It is realized previously that enzymes can hasten the chemical reactions, and that is they can make the reaction go faster by 5 to 17 orders of magnitude. The question here is how and also why. What is the source of the energy for lowering the activation energy? So there are two mechanisms involved. Number one is rearrangement of covalent bonds, forming transient covalent bonds. And then this allows transient transferring of chemical group from the substrate going to the enzyme, and it usually happens on the active site. But the more common mechanism is non-covalent interaction. So this non-covalent interactions are made up of weak forces, and these are the forces also similar that stabilize protein structure like hydrogen bonds and also van der Waals interaction. In other words, these non-covalent interactions is or are collectively called as the binding energy. Na-discuss ko na to kanina. This is the energy derived from energy substrate interaction, and it is the difference between the activation energy of uncatalyzed reaction and also catalyzed. Ay, na-discuss natin siya kanina. And this serves as the major source of free energy used by enzymes to be able to lower the activation energy. So later we will get back to that sa isang figure. Next, we have specificity. Enzymes are very specific. That means they can discriminate substrate na almost similar. Kung sino ba talaga ang para sa kanila. Okay? In relation to this, we have induced fit model and also lock and key hypothesis. In induced fit theory, it means that when an enzyme or when a substrate binds to the active site of the enzyme will undergo conformational change. So mag-adjust siya to be able to make the binding suitable between the substrate and also the enzyme. This conformational change is said to be analogous in placing a hand into a glove that is suited or that is matched. Ay nagkakaroon ng conformational change. So kumbaga, pwede nating i-connect dito yung magkarelasyon na nag-break and nag-promise yung isa na hindi na niya re-replyan yung message nung kanyang ex. Halimbawa, siya yung enzyme. Ngayon, nung nag-text yung kanyang ex, hindi niya nagampanan yung sinabi niya na magiging rigid siya. So, nagkaroon ng conformational change, nireplyan niya. Okay? Another is lock and key theory. So, this was first postulated in 1894, in which lock represents the enzyme and the key represents the substrate. And kapag may perfect match ang lock and key or the enzyme and also the substrate respectively, there is correct fitting resulting to a product formation. And it is said that lock and key hypothesis provides perfect fitting between an enzyme and also the substrate. However, this information could be misleading because take note that kapag totally complementary ang enzyme at ang substrate, that enzyme is said to be a poor enzyme. Instead, to make it a good enzyme, hindi dapat perfect fit. Kailangan magkaroon ng proper interactions between the enzyme and the substrate that would happen only in the transition state. So, kailangan ma-achieve muna ang transition state bago magkaroon ng perfect match, hindi right away. So, to be able to explain that, let's have this sample analogy. Huwag niyo munang pansinin yung graph sa kanan, ito munang nasa kaliwa. Halimbawa, ang analogy natin dito is meron tayong metal stick. Ito halimbawa yung substrate. So, ang goal natin sa metal stick, gawin siyang product in which puputulin siya, ay puputulin. So, ang transition state para maputol ito, kailangan mag-bend, ay mag-bend yung stick. So, through bending, that's the molecular moment. Yan yung climax na kung saan hindi natin alam kung matutuluyan ba ang metal stick na maputol o hindi. So, para maputol siya, kailangan nga ng greater energy than the transition state to overcome. So let's say we have the second scenario in which ang enzyme ay totally perfect fit doon sa substrate. Complementary siya right away doon sa substrate. So kapag may perfect fitting, few products lang ang nagagawa kasi it causes initially lowering of the Gibbs free energy when enzyme and substrate was formed. So ang tendency dito is malaki yung activation energy na makokonsume, and higher activation energy would mean slower reaction. Whereas kung ang perfect fit ay mangyayari sa transition state, so for example, at first, nung hindi pa na-reach ang transition state, hindi pa siya perfect match or complementary. Nung nagkaroon ng transition state, nung nag-bend initially yung metal stick, may perfect fit na nakuha lang ng transition state. Ang pinagkaiba ng C sa B, nakuha ang perfect match kapag transition state na, yung kanina hindi pa. So, ang tendency, bababa ang activation energy. So, ang activation energy, pag mababa, mabilis ang reaction. Kaya, according to other studies, mas okay na perfect fit at transition state, not kapag ground state pa, not during ground state, not during ground state. Notice that the color green, not here, this one is the binding energy. That's the difference between the uncatalyzed and catalyzed energy. So ito na yung diniscuss natin kanina, binding energy between uncatalyzed and catalyzed. Okay na yan. This is the activation energy of uncatalyzed and this is the activation energy of catalyzed. Ano sabi natin? Pag mataas ang activation energy, mababa ang binding energy. Pag mababa ang binding energy, slower ang reaction. Okay? Pero pag mababa ang activation energy, mataas ang binding energy, kaya ang reaction ay yes, very good, faster. Okay, so yun. So I hope you can follow. Saan na ba tayo natapos? Dito, mechanism. So for the different mechanisms to facilitate catalysis, number one, we have acid-base catalysis. This refers to proton transfer because of the presence of acids and bases. Transferring of electrons naman for covalent catalysis, and metal ion catalysis in which there are ionic interactions. So for instance, the one that provides the three mechanisms or combination is the enzyme chymotrypsin, having both covalent, acid-base, and transition state. So we have different factors affecting enzyme concentration. For now, please take this note muna kasi babalikan natin to pagpunta natin sa second part ng enzyme kinetics. So babalikan natin to pagpunta natin ng part 2, enzyme kinetics. So substrate concentration, stay tuned, babalikan natin to, ulitin ko, temperature and pH. So video, we tackled enzymes, their actions, and their ability to lower the activation energy. At this juncture, we are about to lecture enzyme kinetics. Now, biochemists use several approaches in studying the mechanism of action of enzymes. And one of the oldest and essential methods in understanding the MOA of enzymes is by determining the rate of reaction and how it changes in response to changes in experimental parameters. And that discipline is known as the enzyme kinetics. This is the field of biochemistry that is concerned with the quantitative measurement of the rates of enzyme catalyzed reactions and the systematic study of the factors that affect these rates. In enzyme kinetics, this is the approach being applied so as to understand the MOA of enzymes or their ability to convert the reactant or the substrate into a product. And in having enzyme kinetics, this is usually coupled with experimentation. And the data being obtained in experiment is usually plotted using a graph or using a plane. In particular, on the vertical or y-axis, we have here the reaction velocity, and on the x-axis, we have the substrate concentration. So when we talk of reaction velocity, this is also known as V naught, and this is defined as the number of substrate being converted into a product per unit time. Usually, the unit is millimoles per unit time. That's for V naught, okay? And then for the substrate concentration, that is, of course, the amount of the substrate that is expected to be converted to a product through the presence of an enzyme, okay? And then the dashed lines presented is called as the Vmax, okay? Or the maximum velocity, okay? Maximum velocity. Now, ano usually ang graph na makikita natin kapag pinlot natin ang mga data, kapag ginamit natin ang mga values na ito? So usually, ang setting in experimentation for enzyme kinetics is that the concentration of the enzyme is held constant. Again, the concentration of the enzyme is held constant. So meaning, same amount lang ng enzyme ang ginagamit that would react to the substrate. Whereas, ang concentration naman ng substrate, ito yung variable. Okay, ito yung variable. So ito yung variable, meaning ito yung nagbabago. Okay? In particular, pataas ng pataas. So ano yung observe graph natin kapag tinest natin ang isang substrate with an enzyme that is constant in value. Take note at low, again at low substrate concentration, again at low substrate concentration, V naught, again V naught increases, it increases almost linearly, linearly with an increase in the substrate concentration. So at low substrate concentration, there is almost linear, a linear graph that can be observed as the substrate concentration increases. However, if we further increase the substrate concentration, you can see that V naught increases by smaller and smaller amount. Okay? Habang tumataas ang substrate concentration further, lumiliit na lang ng lumiliit ang changes sa V naught. Okay? Changes sa V naught. And at even higher substrate concentration, you can see that V naught almost reaches the Vmax asymptotically. Pag sinabi nating asymptotically, it's too close but it never intersects with the Vmax. Okay? Asymptotic yung graph. So that's what can be seen on the graph kapag sa enzyme kinetics with constant enzyme and varying substrate concentration. And pag na-reach natin ang asymptote o yung plateau-like region, that means na-reach na natin ang maximum amount ng substrate na naging product o magiging product. Okay? And ang itsura ng graph like this is characterized by having hyperbolic graph, a hyperbolic graph. So kapag hyperbolic graph ang nakita natin, that explains Michaelis-Menten kinetics. So Michaelis-Menten kinetics is the relationship, is the way, or is the principle that explains quantitatively the relationship between the substrate concentration and the reaction rate. So it states that the enzyme first combines reversibly with its substrate in a fast reversible step, and kapag na-form ang enzyme-substrate complex, so for example, this is the enzyme and this is the substrate, so the rate of reaction forming ES or enzyme-substrate complex is a fast process and it's reversible. But the dissociation of the enzyme-substrate complex forming finally the product is a slow process. And in Michaelis-Menten equation or kinetics, to be able to derive the equation on Michaelis-Menten, the following assumptions were made. Number one, the substrate concentration is always greater than the enzyme concentration, so that the enzyme substrate concentration at any time is small. So since lagi mataas ang concentration ng substrate kaysa sa enzyme, ma-reach of course ang maximum amount ng substrate na magiging product lang, kasi hindi lahat ma-occupy ng ma-accommodate rather ng enzyme. And in steady state assumption, that means pwede ma-reach ang steady state, and it means the rate of formation of ES, again the rate of formation of ES is equal to the rate of breakdown. Again, that is equal to the rate of breakdown. So when we say steady state, it means that the rate of enzyme substrate formation is equal to the rate of degradation or breaking down. Okay? And initial velocity is used in the analysis of enzyme reaction. Remember, ang initial velocity is also the V naught. Okay? V naught. And in V naught, inaalam natin dito ang rate of enzyme catalyzed reaction. Gaano karami ang mabubuong enzyme substrate complex na ready ultimately maging product. Okay? So this is used in the analysis of enzyme reactions, and the rate of reaction is measured as soon as E and S are mixed, forming enzyme substrate complex. As we said, kapag na-form ang enzyme substrate complex, mapafacilitate ultimately ang formation ng product. Okay? So product concentration is very small and the rate of back reaction can be ignored. That's why the K-2 can be ignored already. Okay? So, based on those assumptions, this is finally the Michaelis-Menten kinetics. So, we have V naught, Michaelis-Menten equation. So, we have V naught is equal to Vmax multiplied by the substrate concentration all over Km plus substrate concentration, where V naught is the initial velocity. Again, that is the rate of enzyme catalyzed reaction. A rate of enzyme catalyzed reaction. Then maximum velocity, the one that is on the dash, the one with dashed lines that were presented earlier, and then initial substrate concentration found on the x-axis, and the Michaelis constant. So when we say Michaelis constant, this is actually the substrate concentration. Again, when we say Michaelis-Menten constant, this is defined as the substrate concentration at which V naught is 1/2 of Vmax. Again, when we say Michaelis constant, this is defined as the substrate concentration at which V naught is 1/2 of the Vmax. So with that, let's check the graph that was shown earlier. So in Michaelis-Menten kinetics, the initial velocity or V naught in micromolar per minute is usually seen on the vertical or y-axis. Substrate concentration in micromolar, that is in x-axis, and then we have Vmax here. As you can see, plateau-like graph could be observed here, signifying that the maximum enzyme has been utilized. And ang characteristic ng graph like this is asymptotic. And yung overall graph is hyperbolic, right? Now, ang nadagdag is Km. Sabi natin Km is the substrate concentration. This is the substrate concentration at which ang V naught ay 1/2 ng Vmax. Okay? So 1/2 ng Vmax. So Km again is the concentration at which V naught is 1/2 of the Vmax. Now question is, what is the importance of Km? Again, what's the importance of Km? That will be discussed later pag proceed natin sa mga conclusion. So tandaan nyo ang Km, ito yung amount or yung concentration ng substrate na kung saan nakalahati na ang Vmax. Okay? Next, for the Michaelis-Menten kinetics, Vmax is observed when virtually all the enzyme is present as the enzyme substrate complex and concentration of enzyme is vanishingly small. In other words, pag na-reach ang Vmax, that means ang enzyme is already saturated, or in Tagalog, puno na siya. Hindi na niya kaya mag-accommodate sa kanyang active site ng iba pang mga substrate para gawing product. So that implies saturated na siya. And saturation of enzyme can be seen when plateau is observed on the graph. Okay? So it means hindi na ma-accommodate yung ibang substrate. Kaya nga nabanggit natin kanina, sa umpisa, linear siya at low substrate concentration. So sa umpisa at low substrate concentration, almost linear kasi marami pang enzymes available. Pero pataas ng pataas ang substrate concentration, pero yung enzyme constant. Kaya hindi na ganoon kataas ang nako-convert into product hanggang hindi na maging available yung enzyme. And for the conclusions of Michaelis-Menten, unahin natin ang Km. Again, unahin natin ang Km. Recall class na ang Km, this is the substrate concentration na kung saan kalahati ng V naught or kalahati ng Vmax ay nakuha na. So sinabi natin kanina na pag na-reach ang Vmax, that means saturated na ang enzyme. So altogether, pag sinabing Km, ito yung dami ng substrate na kailangan upang yung kalahati ng saturation point ay ma-achieve. Okay? So may implication yan depende kung malaki or maliit ang Km na nakuha. Number 1, small Km. Pag sinabing small Km, sa graph niyan, it's near 0. So pag sinabing near 0, that means low Km value, small Km. So pag small Km, that means you only need small amount of the substrate para makalahati ang saturation. It means sobrang attracted ng substrate doon sa enzyme. Kaya small km signifies high affinity of the substrate to the enzyme. Okay? And pag large km naman, it means it takes so much, it takes a high amount of the substrate needed para ma-half saturate ang enzyme, signifying low affinity. So ulitin natin, pag small km, high affinity, pag pag low km high affinity, again pag low km high affinity, pag high km low affinity. Next, relationship of velocity to enzyme concentration. The rate of reaction is directly proportional to the enzyme concentration at all substrate concentration. So it's directly proportional. And sa order of reaction, it's first order if the velocity or the V naught is directly proportional to the substrate concentration. Whereas pag zero order naman, constant. Pag zero order, it's said to be constant. After you learned what Michaelis-Menten equation and graph look like, let's have the main limitation of Michaelis-Menten equation or graph. Remember that the graph that can be observed is a hyperbolic graph, and the possible problem here is the graph does not really touch the Vmax. It's only asymptote. So ang implication nun is kapag Michaelis-Menten ang graph or hyperbolic ang graph, you cannot actually determine the exact value of Vmax because it doesn't touch at all. And when you cannot get the exact value of Vmax graphically, you cannot determine Km after all. Kasi ang Km is half concentration needed para makuha ang half ng Vmax. So, pag hindi mo nalaman ang Vmax, hindi mo malalaman yung Km. Kaya, problem ito. So, to be able to address this issue, we have to convert this graph into a linear graph. And to be able to make it happen, we have to apply the principle of slope-intercept form, a slope-intercept form. In algebra, remember that the slope-intercept form or equation is like this: y is equal to mx plus b. So by converting this graph into a slope-intercept form, we can get a linear or straight line. And by having the straight line, we can get the exact value of Vmax. And to address this issue, to get the slope-intercept form, we have to double reciprocal the Michaelis-Menten equation. So pag sinabi nating double reciprocal, i-reciprocal natin ang left and right side of the Michaelis-Menten equation. So ang tawag doon ay double reciprocal equation. And ang tawag ultimately doon sa equation na double reciprocal is Lineweaver-Burk equation. Ulitin natin, Lineweaver-Burk equation is also known as double reciprocal until you get this equation. So this is the Lineweaver-Burk equation. So how does this relate? How does this relate to the slope-intercept form? 1 over V naught represents the y, y, and then Km over Vmax, Km over Vmax is the slope. Again, Km over Vmax is the slope. And then 1 over substrate concentration. This is also equivalent to this, right? So 1 over substrate concentration is the value of x. Okay? Slope is m, right? mx. And then plus 1 over Vmax is b. B is the y-intercept. Okay? This is the y-intercept. So anong ibig sabihin natin ng y-intercept? So y-intercept is defined as the value of y when x is equal to 0. Okay, again, this is the value of y when x is 0. So in other words, ang y-intercept, ito yung value ng y na nag-intersect. Okay, this is the line or value of y at the point where the line crosses the y-axis, nag-intersect siya sa y-axis. Okay? Kabaliktaran siya ng x-intercept because ang x-intercept naman is the value of x when y is equal to 0. Again, x-intercept is the value of x when y is equal to 0. So that represents y-intercept 1 over Vmax. And pag x-intercept naman, that is 1 over substrate concentration. Again, that is 1 over substrate concentration. So by converting the Michaelis-Menten equation to Lineweaver-Burk or double reciprocal equation, we can obtain this graph which is linear. So remember, 1 over V naught is the y, a value of y, or y equivalent, and then Km over V max is the slope, m ito, m, and then we have 1 over substrate concentration, na-overlook ko, this is not 1 over substrate, but rather 1 over Km, ay 1 over Km, yung x-intercept natin, that is in negative symbol, ay 1 over Km, negative symbol. So, paki-modify na lang class, it's negative 1 over Km, and then, this is the value of x, and then y-intercept, this is 1 over Vmax. This is the y-intercept, and ang x-intercept, negative 1 over Km. Notice that the y-intercept is the value of y na nag-intersect sa y-axis, and that is the value of y where x is 0. And same thing with x-intercept, this is the value of x when y is 0 naman. Okay? If you can observe the y-intercept or the 1 over Vmax, na-intersect na niya ang graph, ang line. So therefore, may exact value tayo ng Vmax, only in reciprocal. Pero at least, makukuha natin exact value. That's why you can get also the Km in turn. Okay? So yan ang yan yung solution natin doon sa problem na asymptotic pag Michaelis-Menten. Okay? Moving on, let's proceed on, dito na tayo, different types of reversible inhibition. So there are three different types of reversible inhibition. These are competitive. Second, we have uncompetitive. And lastly, non-competitive inhibition. So first stop, we have competitive. When we say competitive inhibition, this means that the substrate and the inhibitor, again, the substrate and the inhibitor compete at the same active site of the enzyme. So say for example, on the figure, we have the substrate and the inhibitor, and this is the active site. So this compounds or these entities compete for the same active site. However, you have to realize that inhibitors, competitive inhibitors, are designed to have greater affinity. They are designed to have greater affinity than the substrates themselves. That's why the one that will bind on the active site of the enzyme is the inhibitor per se. Kaya, ang magbabind inhibitor, substrate hindi. So, ano ang dapat gawin para ma-counteract natin o para ma-kick off, para maalis ang initially bound inhibitor? The solution in this problem is we have to increase the amount or concentration of the substrate than the inhibitor, because the higher the amount, the greater the collision between the substrate and the enzyme, thus displacing or kicking off the inhibitor that is initially bound to the enzyme. Okay? So how does this reflect on the Lineweaver-Burk plot? Okay? Lineweaver-Burk plot. So tandaan natin sa competitive inhibition, since pinaparami natin ang substrate concentration, affected dito ang Km, okay, Km. So that means bumababa ang Km, kapag merong competitive inhibition. So tumataas ang Km because we need greater concentration of the substrate. So tumataas yung Km. Pero ang Vmax ay equal or unchanged. Ay unchanged ang Vmax. Again, Km is being increased because we need higher concentration, higher amount of the substrate, and higher amount of the substrate will move the Michaelis-Menten graph to the right, kaya tumataas siya, and then unchanged naman sa Vmax. So mamaya papakita natin yung graph. Okay? Next, for the example, we have statins. Statins competitively inhibit HMG-CoA reductase. So meaning to say, ang statins at ang HMG-CoA, they compete HMG-CoA for the same active site, which is the reductase enzyme. Between the two, statins are designed to have greater affinity. That's why they will block the activity of the enzyme. Next, we have uncompetitive inhibition. In uncompetitive inhibition, an uncompetitive inhibitor binds at the active site distinct from the substrate active site. So, this means na ang uncompetitive inhibitor, dumidikit siya hindi sa primary active site, rather on the secondary active site. How does it happen? So, for example, this is the enzyme having the active site, and this is the substrate. So, usually, kung makikita nyo, wala pang ibang active site than the primary one. So, substrate binds initially there. And after binding of the substrate on the active site, that creates another groove, another active site or the secondary active site that welcomes or that accommodates binding of the inhibitor. So that's why nakalagay dito, ang inhibitor ay magbabind lang kapag na-form na ang enzyme-substrate complex, because ES complex formation facilitates formation of another active site that's for the inhibitor. And binding of the inhibitor causes formation of the enzyme substrate inhibitor complex that would block ultimately the action of the enzyme. Okay? Sad thing about uncompetitive inhibition, we cannot counteract the effect of the inhibitor already. Kaya ang tendency, bumababa ang Vmax, and then ang problem din dito or yung characteristic niya is binding of inhibitor to the active site causes the substrate to bind further on the enzyme. So pag mas maraming substrate ang nagbind doon sa enzyme, more ES complex. And more ES complex signifies that more secondary active site could be formed, thus increasing binding of the inhibitor. So the more the substrate that binds on the active site, the more grooves that could be formed. So anong implication pag maraming substrate ang dumidikit? Greater affinity, di ba? Greater affinity, mas na-attract yung substrate doon sa enzyme. And greater affinity tells us na low Km. So dito bumababa ang Km. Next, what about non-competitive inhibition? Again, paano naman kapag non-competitive inhibition? So sa non-competitive, this is rarely encountered in experiments, and a mixed inhibitor also binds at a site distinct from the substrate active site, but it binds either to the enzyme per se or the enzyme substrate complex. Now the problem here is ang Vmax ay bumababa, and ang Km ay unaffected. Km is unaffected. So Vmax is reduced and Km is unaffected. So these are the graphs that can be observed kapag may competitive, uncompetitive, and non-competitive. Sa competitive, we said that Km is being increased, Vmax is unaffected. That's why you can see, for example, this is the graph of without inhibitor. Now, nabanggit natin na kapag Lineweaver-Burk nire-reciprocal, di ba? So, pag sinabi natin Km ay increase, sa Michaelis-Menten, lalayo siya sa graph, usually, pag Michaelis-Menten. Ngayon, pag nire-reciprocal mo yung mataas na Km value, magiging kabaliktaran siya instead of malayo, lalapit. Kaya yung negative 1 over Km niya, it's near the origin. Kaya dito siya. And ang Vmax is unaffected, kaya nandito lang din siya, intersecting line ngayon ang mangyayari. Okay? So, ang kabaliktaran ng lumaki, lumiit. Kaya maliit siya dito, double reciprocal. Then, pag uncompetitive naman, Km is reduced. So, Km is reduced and Vmax is reduced. So, that means, kung reduce ang Km, kung maliit siya sa Michaelis-Menten, lalaki siya pag Lineweaver-Burk kasi reciprocal kabaliktaran, di ba? Kaya, from no inhibitor, again, from the graph without
without the inhibitor. Pag meron ng inhibitor, you can see na ang negative 1 over Km, yung reciprocal form ng Km reduced, lumayo siya sa 0. Nandito na siya. Okay? And then, kapag naman Vmax is reduced, so kung sa Michaelis-Menten, bumaba ang Vmax, pag binaliktad mo yun, lumayo ang Vmax. Kaya nandito siya. From here, napunta dito. So, lumayo. Kaya parallel line siya. And then, pag non-competitive naman, Km is unaffected. Km is unaffected. Kaya pansin nyo, same value siya dito. And then Vmax is reduced. Ano sabi natin? Pag Vmax is reduced sa Michaelis-Menten, kapag Lineweaver-Burk, lalayo. Kaya nandito siya. Okay? So, yun yung mangyayari. Next. So, yung mga susunod na slides, actually self-explanatory na lang sila. Dito sa allosteric enzyme, ang tandaan nyo lang, class, is pag Michaelis-Menten, di ba, right? It's hyperbolic. Pero pag allosteric, it's sigmoidal. Represented by the color green. May color green na graph. Sigmoidal siya. So, dito na tayo sa enzymes in clinical use. Just take note of the names of the drug and the enzymes that they block. Actually, ito yung pinagawa ko sa inyong activity last time, na may mga gamot tayo that can target enzymes that would have beneficial effects or clinical uses. And the principal serum enzymes used in clinical diagnosis are the following. We have aminotransferases, amylases, ceruloplasmin, creatine kinase, glutamyl transferase, lactate dehydrogenase. Next, lipases and phosphatases.